Greetings, I am Norsyasya Adriana from UPM. Will be presenting my research entitled Elucidation of Molecular Biological Mechanism of Gemcitabine against Pancreatic Cancer via Integrated Bioinformatics Analysis. Pancreatic cancer is mainly considered an incurable disease. It has contributed to a poor prognosis in cancer diagnosis since it is often detected at the later stage of the disease. Since 1997, gemcitabine has replaced fluorouracils as the standard first-line drug for advanced pancreatic cancer. However, to date, the 5-year survival rate of a pancreatic cancer patient has not yet improved, and gemcitabine treatment alone often associated with the resistant development compared to gemcitabine combination-based therapy. So this current study aimed to explore the key gene targets, basic pharmacological effects, and molecular biological mechanism of gemcitabine against pancreatic cancer by conducting bioinformatic analysis. So the methodology for my study consisting of 3 steps, which are target identification, screening potential target, and network analysis. For target identification, potential gene targets of gemcitabine were predicted through the drug bank database, disease gene interaction database, and comparative toxicogenomic database, while pancreatic cancer gene were predicted through Malachite and online Mendelian inheritance in man databases. The intersection gene between gemcitabine and pancreatic cancer were then created using the online Venn diagram tool to screen for pancreatic cancer-related target genes of gemcitabine. Then after that, pancreatic cancer-related target genes were imported to string database to construct protein-protein interaction network and find the hub genes. Cytoscape and David bioinformatic tools were then used to construct compound target pathway network and conduct the enrichment analysis respectively. As a result, 352 potential gene targets of gemcitabine were predicted with 89 shared with pancreatic cancer. These shared genes could act as functional target in pancreatic cancer and are used for further analysis. The PPI network presented as figure 1 here showed that the shared target genes have complex interaction between each other. The network contained 88 nodes with 1 disconnected node of gene, MIR242, was hidden and 425 edges. The nodes, the circle in the figure, represent the target genes, while the edges, the line, represent the interaction between them. From the PPI network, several genes which are TP53, EGFR, AKT1, VEGFA, CCND1, MYC, and STAT3 listed in Table 1 have been identified as the hub gene based on degree, closeness, and bituminous value. Additionally, there are 3 main dysregulated genes other than TP53 in pancreatic cancer targeted by gemcitabine, which are KRAS, CDKN2A, and SMART4 presented in Table 2 with their respective degree, closeness, and bituminous value. The possible biological process and pathway in the treatment of pancreatic cancer was further evaluated through enrichment analysis. In the gene ontology enrichment analysis, the most enriched biological process are negative regulation of apoptotic process and positive regulation of cell proliferation. It has been reported that failure in activating the apoptotic process and abnormal proliferation of cells could lead to cancer. Therefore, by targeting the genes involved in the process that prevent apoptotic process and increase cell proliferation, the cancer cells will probably reduce in number, thus changing a pancreatic cancer to a curable disease. In the CAP pathway enrichment analysis, the most enriched pathway involved are mostly cancer-related pathway, including pancreatic cancer. Next, figure 3. The compound target pathway network using cytoscape demonstrated that gemcitabine exerted its therapeutic effect by regulating the top 10 pathway with the involvement of multiple targets. The network contained 100 nodes and 2,000 edges, including 1 compound, 89 target genes, and 10 pathways that constitute the network node. This compound produced a total of 2,000 interactions with the target genes and also the pathway. As for discussion, TP53, EGFR, AKT1, VEGFA, CCND1, MYC, and STAT3 were recognized as the hub gene in this study and highly interconnected with nodes in the network and possess high biological importance in a certain disease. Next, the main dysregulated gene in pancreatic cancer, which are TP53, KRAS, CDKN2A, SMART4, were found to be targeted by gemcitabine as well, but only TP53 recognized as the hub genes. Our in silico study correlate with the previous study where gemcitabine mainly act on several cancer-related pathways, including pancreatic cancer, through regulation of apoptotic process and cell proliferation. To conclude, this study may provide the insight to explore gemcitabine resistance and enhancing its effectiveness with other drugs like nab-paclitaxel or bioactive compounds for future use. Below here are the references. And with that, I would like to thank all of you for listening to my presentation. Thank you.